This is session 6, muscle protein turnover and translational control of muscle mass. We have 4 speakers in this symposium. Just want to start by saying thanks to Dave Hood and the crew at York for doing such a fantastic job, and all the sponsors that you can see up on the slide here for making IBEC possible. 4 speakers: Dr. Marco Sandri from the University of Padua, Sue Bodine from the University of Iowa, Troy Hornberger from the University of Wisconsin, and then saving the worst for last, I get to speak. So without further ado, if I could ask Dr. Sandri to come up to the podium and talk about novel pathways in muscle mass regulation, the role of mitochondria.
So thank you for David and the organizer to give the possibility to present our data at this meeting. And today I want to talk a story in which we link mitochondria with the secretome of the muscle and how this can affect, in catabolic condition, affect muscle loss. I guess that everyone is aware in this audience how deleterious can be muscle loss in different conditions, like if you have cancer, heart failure, infection, lung disease, but also kidney and liver disease can lead to a massive muscle loss. It's called cachexia. Just in cancer, 30% of the people that die for just cachexia, not for the cancer. So one question I have in my lab is why cachexia happens in cancer. And we start from humans, so we have up now more than 400 biopsies that were collected from people that have esophagus, pancreas, colorectal cancer, and controls. And we have a biopsy from people that have cachexia, but they have also the experience, the muscle loss or weight loss, and they are not yet cachectic. So we have also the condition which have a pre-cachectic situation. And when we look at morphology of the histology of muscle cross-section, and you appreciate that the fiber size are smaller, but at the same time, you can appreciate that there are some tiny, small, flat, angulated fibers that are not regenerating fiber. These are, for a pathologist, are denervated fibers. So we start to think that maybe during cancer, we do have alteration of innervation, and maybe the innervation can happen. So just how we can monitor in histology the innervation or the innervated fiber, we just use this marker, and CAM is a protein that is localized at the neuromuscular junction. And when a fiber starts to lose innervation, the protein starts to be relocalized on all the surface of the muscle fiber. So just looking localization all around the fiber or just in a single area, we can catch up with the denervated fibers. So when we look at histology of biopsy from control and cachectic people, now we start to see this angulated or small flat fiber that become positive for CAM, suggesting that indeed alteration in innervation seems to occur during cancer growth. And that happens almost in the pre-cachectic condition. So this is quantification. So before cachexia onset, so you have signs of denervation. We just further want to use more signs of denervation, and we check some by gene expression, some markers, some genes that are induced during when fiber denervated, like myogenin or RANKS1. And again, you start to see a denervation features or profile in pre-cachectic people that experience not cachexia yet. And then we look in the blood, so when you have neuromuscular junction remodeling, some proteins are able to cleave some proteins and release from the neuromuscular junction or the synapse, go into the blood. And this is soluble agrin and soluble CAM, so these are in the blood of the people that have cancer, and you can appreciate that again. Pre-cachectic person, they have already a high level of markers of synaptic degradation. So to have a better morphology of going on of what happens in our muscular junction, we move to animal model cancer cachexia, and we stain the muscle for postsynaptic side here by bungarotoxin, and then you can appreciate the nerve the axon in red, and the presynaptic side with different markers, neurofilament and synaptic vesicle 2. So you have a good match between the fiber innervation and the axon and the nerve in control animals, but in cachectic animals that have a C26 colon cancer, now you appreciate that there are some neuromuscular junctions that are positive or green, but they lack the nerves and the axons. This is morphology. We want to see if a functional assay are affected, so we use electromyography. So this is typical biphasic depolarization event. So you stimulate the nerve and you measure depolarization, repolarization events called compound muscle action potential, CMAP. And if you repeatedly stimulate the nerve, you can have almost no decay in the CMAP amplitude, or a little bit of fatigue, so a decrease in release of acetylcholine receptor is minor. But if you have cancer in animals, now you have this polyphasic CMAP, and you can appreciate the amplitude, the voltage is dramatically reduced, and now you have a decay in the CMAP amplitude, altogether suggesting that indeed, besides morphology alteration, there's a functional impairment in neuromuscular junction when animals has a cancer, tumor growth. So the question is how cancer may affect neuromuscular junction, which are the signaling coming from the muscle go to the motor neurons. And now we jump to mitochondria. Indeed, Teresa Zimmer and Andrea Bonetto did a proteomic analysis on animals that have cancer cachexia, and the major hit that came out is mitochondria. Mitochondria protein dramatically decrease in cachectic muscle. And among the different mitochondria, I want to focus on the mitochondria protein important for mitochondrial fusion and fission, so dynamics, OPA1, multifusin 2, and GLP-1. And you can appreciate they are all downregulated in animal models of cachexia. But even in chemotherapy, when you treat animals with chemotherapy drug, you have myopathy and alteration in mitochondrial function. And again, OPA1 and multifusin 2 and GLP-1, fusion and fission protein are downregulated. And this has been already reported by different groups in other models. So in sarcopenia, this is a transcontinental analysis, transcriptomic analysis of sarcopenic people, and the mitochondria are greatly affected. They have downregulation of most of the genes related at OXPHOS, TCA cycle, but also protein infusion like OPA1 and in fission like GLP-1 or FIS1. And we are involved in a human study, a bed rest study. So this is 10 subjects, they were pretty young, they were bedridden for 10 days, and we can isolate a single fiber and monitor the proteomic on the single fiber and check which protein is going up or down after 10 days of bed rest. There is a short bed rest because the focus is understanding what's happening in the early phase, before you get muscle loss. Indeed, these people, they experience a little bit of muscle loss, but mainly they experience weakness, 25% of decrease in force generation. And we have also muscle biopsy from space flight, but I want to focus just the data about the bed rest. So a short time of inactivity in young people. So surprising, well, of course, the proteins, there are not too much changes. So the significant proteins that go up or down were few, not too many. And the proteins were going up were from the proteasome, subunit, of course, protein degradation occurs. And we listened yesterday from Michael Jackson, the peroxyredoxin, the antioxidant. Indeed, peroxyredoxin 2, peroxyredoxin 1, SOD1 are induced, suggesting that probably there is an oxidative stress going on in this muscle. But importantly, myosin and actin, there's only a minor change in the content. What greatly was changing were costomer and Z-line proteins, so protein belonging to the dystrophin glycoprotein complex, like sarcoglycan, protein of the desmosome, desmoplakin, desmoglein, placoglobin, and protein that are of the Z-line. And I want to focus your attention on these two proteins, CYP1 and CYP2. These are actin binding proteins that are important to anchor actin to the cytoskeleton, important for the desmosome and adherence junction function. So I will come back at the end of the story. So keep in mind that in bed rest, this protein, CYP1, CYP2 are going down or suppressed. And of course, mitochondrial protein, not too much changes, only a minor decrease in mitochondrial protein, but a significant decrease of multifusin 1, multifusin 2, and OPA1. So decrease in mitochondrial dynamics starts before you have a change in mitochondrial mass and mitochondrial protein content. This is a quantification. So this means that in different conditions, you have a cancer, you have a sarcopenic, you have people that have inactivity. Mitochondrial dynamics seems to be affected. And keep in mind that the mitochondria is not a single molecule, it's a network. It's a 3D reconstruction of how mitochondria are in the muscle. They are extremely well connected. So we think that the mitochondrial function is important. I guess that maybe mitochondrial shape is even more important in order to adapt the muscle to different stress and give the signaling. So I do not want to go further about fusion and fission because the previous talk I already mentioned, but in any case, fusion is mainly driven by mitofusin 1 and 2 and OPA1, fission by GLP-1. To understand the role of fusion and fission, we generated recently a muscle-specific inducible OPA1 knockout animals in which by treating with tamoxifen, we can delete specifically OPA1 only muscle. So we wait the animal become adult, and then we treat it with tamoxifen from 3 months of age, and we look what's happened to the phenotype of the animals. Well, the phenotype of the animals was dramatic. In 3 months, they have a cachexia and they die. So they lose muscle mass almost disappear. This is gastrocnemius. After a few weeks of induction, you have the reduction of 50% of muscle of cross-section area, 50% of the fiber number. White adipose tissue is completely gone. Bone is affected. The trabecular density of the bone is dramatically reduced. And keep in mind, this is a muscle-specific inducible OPA1 deletion. And in the blood, you have a tremendous systemic inflammatory response. So you have upregulation of interleukin-1 beta, interleukin-1 alpha, TNF-alpha, interleukin-6, from 10 to 100-fold increase in the blood. So this makes I wonder if can mitochondrial network in some way affect and impact neuromuscular junction, which could be the secret or the hormones or the cytokine that impact the neuromuscular junction. So we did a transcriptomic analysis, and number 1 hit was a cytokine, FGF21, I will come back later, but the number 25 was noggin. It was induced 20-fold when OPA1 and mitochondria were affected. What is noggin? Noggin is a cytokine that blocks the BMP pathway, and we recently found that in this pathway it can promote atrophy. And when we look in cancer cachexia animal model, noggin was induced, as well as in humans that have a precachectic or cachectic condition, suggesting that maybe this noggin is some way can relate it to neuromuscular junction alteration. And we have another animal model that have a muscle-specific autophagy deficient that has a problem in neuromuscular junction, leading to denervation and premature death. And even in this condition, you can appreciate the alteration in CMAP, so polyphasic, and actually in this mice, you have spontaneous depolarization because you have a change in ion channels, so the muscle undergoes spontaneous contraction that is typical of the nervated muscle. And indeed, also in this mice, noggin is induced. And we have also muscle biopsy from our old sarcopenic people, and also in sarcopenic people in which denervation happens, noggin is induced. So it could be that the mitochondrial dysfunctions trigger noggin, and noggin could be the culprit for neuromuscular junction alteration. So if it's true, what's happened? So keep in mind the noggin blocks BMP pathway. BMP pathway is important for protein synthesis, and to block protein degradation, mainly affected the ubiquitin ligase, the muscle-specific, but also a novel ubiquitin ligase that we discovered in 2013. It was called MUSA1. And indeed, when you express noggin in a muscle, you can appreciate how much you have the shrinkage as a global muscle mass. So what's happened if we overexpress noggin to the neuromuscular junction in muscle? So this is now I change the color, so now the postsynaptic side is in red, the presynaptic side is in green, the axon and the nerve you can appreciate. But when we overexpress noggin, now you complete again. You have a retraction of the axons, and only the postsynaptic side is present in the muscle fiber, confirming the denervation. And indeed, when we check electromyography, the traces, again, this is a normal biphasic depolarization event, but when you overexpress noggin, you have this polyphasic. And now, again, normal depolarization events happen, confirming that noggin overexpression in muscle is sufficient to dismantle neuromuscular junction and reduce innervation and trigger the denervation. So if it is true, by restoring BMP pathway in cancer and tumor-bearing mice, we should be able to prevent muscle loss and prevent neuromuscular junction alteration. That was an experiment we overexpressed with either BMP7 or type 1 receptor, L3, And now we partially or totally restore muscle mass in tibialis and gastrocnemius of cancer or tumor-bearing mice. What about the electromyographic traces? So this is the polyphasic and the decay in amplitude that happens when you have a cancer in animal model. But when we overexpress BMP7, now we restore a biphasic CMAP, and now the decay does not happen anymore, either when we express BMP7 or DIPE1 receptor actually suggested indeed BMP, if you maintain BMP pathway, you maintain a muscular junction even in catabolic condition, and you maintain neurotransmission. If you don't have BMP, now you start to get in trouble with innervation in muscle fiber. So the question is, why noggin is induced in cancer and in condition of mitochondrial dysfunction? So we compare two models in which have cancer, colon cancer like MC38 that does not induce cachexia, and C26 cancer that induce cachexia and be implanted in mice, and we check in the blood different cytokines that can be related to muscle loss. We check activin A, that is one of the most, let's say, triggers of muscle loss, but that was induced both in a cachectic colon cancer, but also non-cachectic colon cancer. What was instead in the blood unique was interleukin-6. So interleukin-6 was only present in tumor-bearing mice that has a colon C26, and they are cachectic. So we started to think about, and interesting enough, this IL-6 expression also increases as a transcript in the muscle. So we think that maybe muscle can contribute to the increase in the blood of IL-6. So we tested that maybe IL-6 is important. We infected muscle with IL-6 overexpressing, but also another pro-inflammatory cytokines, TNF-alpha, and we checked which of these two cytokines can induce noggin. Only IL-6 was able to induce noggin, not another pro-inflammatory cytokine, suggesting that it is a peculiar of IL-6 pathway. And if it's true, this pathway is going through STAT3, and maybe STAT3 is important to upregulate noggin, so we decide to knock down STAT3, and in the context of IL-6 expression, now we do see a reduction in noggin upregulation. So to summarize this first part of the data that has been published, mitochondrial dysfunction happens when mitochondrial dynamics has compromised that can trigger different signaling that can contribute to protein breakdown. At the same time, they are able to induce IL-6 expression from the muscle. IL-6 expression together with other IL-6, maybe coming from the cancer or other organs or other cells, activate a STAT3 pathway that induce noggin expression. Now noggin can prevent BMP pathway, leading to neuromuscular junction instability, increasing protein degradation and inducing weakness and fatigue that is more affected than muscle loss, is more important than muscle loss. So one of the points I have is what is downstream here? So why mitochondria can induce IL-6 expression? So today we already heard in these days about autophagy and mitophagy. So in our cell, all the day, all the time, autophagism are formed around 20 minutes in muscle even faster, and are going through the lysosome, and then the lysosome destroy the cargo. And it is normal physiological process. But there are some conditions in which you have an increase or an overload of the autophagy, an excessive autophagy, like when you overexpress FGF21. This has been shown by Vanina Romanello in this paper. Now, having too much autophagy, mitophagy, it will lead to an overload of lysosome. If you don't have any more lysosomes that are generating, now this lysosome cannot handle all the cargo coming inside. And they can be leaky, maybe for mitochondrial DNA, then can activate a toll-like receptor pathways that induce an FKB, and that can activate a pro-inflammatory response like IL-6 expression, that can contribute to cachectic phenotype. So if it is true that there is an excessive autophagy going on when mitochondrial dysfunction, by reducing autophagism formation, now we should reduce the number of autophagism. Now lysosomes are not anymore overload. Then they do not release any more mitochondrial DNA fragment, and the pathway should be down, as well as IL-6. So we check this hypothesis in our OPA1 deficient animals. We knock down LC3, and we check IL-6 expression, and you can see that now reducing autophagus number, we reduce IL-6 expression in muscle of OPA1. So if the pathway is true, so we know that autophagism seems to be important, is because toll-like receptor number 9 is activated. We decide to knock down, in OPA1 context, toll-like receptor number 9, and now we should have less IL-6 expression in the muscle. And indeed, that was the case. We reduced IL-6 upregulation in OPA1 deficient animal. So if a toll-like receptor is going to an FA-kappa B, by blocking an FA-kappa B, we should be able to now blunt IL-6 expression. And that was the experiment. We overexpress IKB superrepressor, and now we completely blunt IL-6 expression in muscle of OPA1 knockout. So I guess that when we are talking about catabolic signaling, there is not one single hit. There is a lot of signaling pathways coming in which mitochondria can play a role activating different transcription factors that can release different cytokines we can call myokine. And the milieu of the different cytokines can be pro-lipolytic, pro-inflammatory, and as we mentioned, noggin, that can alter neuromuscular junction altogether, leading an enhancement of protein degradation, pro-inflammatory situation that leads to a cachective phenotype with a great weakness and fatigue and exercise intolerance. In the last part of my talk, I want just to focus on the novel ubiquitin ligase we discovered. MUSA1 is induced in cancer cachexia, is induced in disuse, is induced in aging. What is doing? What is the role? Is controlling protein degradation in muscle? To address this important, how much importance is MUSA1 in protein breakdown in muscle? We generate a muscle-specific knockout animals. I just showed you the myosin light chain 1F3 promoter. We have also the inducible muscle-specific. The idea is that blocking MUSA1, maybe we are sparing muscle mass, and maybe we can have a better muscle mass. By looking at young animals, the body weight and lean mass between male and females, there is no any beneficial increase in muscle mass and body weight. We check if we have maybe protection during aging. Maybe MUSA1 can be, since it's induced in aged animal, we can in some way prevent. But actually, we got the opposite result. So we start to have that in males, there's a significant reduction in body weight and in lean mass. And in females, we have a trend, but there is, for sure, a tendency to decrease. And when we look histology, now you start to appreciate there are a lot of heterogeneity, big fiber, but also tiny, small fiber that are very atrophic. And plus, you can see this eosin positive orange-shaped, circle-shaped staining that is typical of maybe protein aggregates. And when we stain for SDH, we do see region of fibers that are completely empty of mitochondria. And this area, when we stain for a marker of protein aggregates, so PCC2 sequestosome 1 is one protein that is able to bind polyubiquitin protein and is able also to trigger protein aggregation because it has a oligomerization domain. You can appreciate that now we start to have inside the fiber in the region that are empty from mitochondria, this positive staining suggesting maybe protein aggregates going on there. To have a better idea, we check the electron microscopy. This is normal muscle with a normal sarcomere and striation and Z-line alignment. Now, when you don't have any more MUSA1, you have this disalignment of the Z-line, and these electron-dense bodies, rod-shaped, you can appreciate the higher magnification, even higher, that are typical of one myopathy. It's called Nemaline myopathy. This is a human muscle biopsy on people that have Nemaline myopathies are mutation in a Z-line protein like actinin or nebulin that are important for the sarcomere integrity, suggesting that maybe MUSA1 is controlling the half-life of the protein with Z-line. And if you don't have box 30, now you start to accumulate that Z-line protein and then form these rod-shaped aggregates. Indeed, these mice are weaker in terms of absolute force and in terms of normalized force. And when we did the proteomics, so the idea is that if this 5-box protein is not working, now the substrate should accumulate. So by doing a proteomic analysis, we should identify which could be the potential substrate for this ubiquitin ligase. And we did a proteomic analysis, and look, now, you remember XIRP1, XIRP2 that were down-regulated during a bed rest study in humans, are Z-line protein able to attach the actin and maintain the cytoskeletal form. We have an accumulation of this protein in MUSA1 deficient animal. We have also PCC sequestosome 1, also LAMP2 suggesting lysosomal dysfunction and autophagy dysfunction. And indeed, just a few examples of some Z-line protein. So it is a muscle from wild type and knockout animals, and we stain for XIRP2, and you can appreciate that they have accumulation in the fiber of XIRP2 that is age-dependent. And this is another protein acting in 3 of the Z line that we identified by the proteomic to be accumulated. And again, you have a tremendous accumulator, is age-dependent inside the muscle fiber. So how we can link all together all this information in a condition of cachexia in which we have activation MUSA1. We believe that MUSA1 is taking care about the dismantle of the Z-line protein. And this maybe is important because this dismantle Z-line protein, now you have more release or more the proteasome or the proteolytic system can be accessed to the sarcomeric protein, the myosin and actin that can be released now and can become soluble and can be degraded by the different proteasome or in case of calpains, caspase, or other kind of proteolytic system. So that's my end of my talk. I have really to say thank you to a very talented group of people in my lab. All the story about neuromuscular junction and cancer cachexia and a human study have been done by Roberta Sartori and the PhD student Camilla Pizzini. All the OPA1 story it has been done by the postdoc Katrina Tetz that's here in the meeting. So if you want to have more lights, there's also a poster about how exercise can help when you have animals that have mitochondrial dysfunction. And I'm done. So I'm taking your question. Thank you for your attention. Sorry, I forgot the collaborators. Sorry. The surgery department for the biopsy we collected, thank you. The people who did with Paul Grigorich all the virus and the BMP story, people for the proteomic, for our MUSA1 knockout, and for the single fiber analysis, Matthias Mann and Marta Murgia in my department. Thank you, sorry.
Thank you very much, Dr. Sandri. This paper is now open for questions. Please.
Really great talk. Always fun to listen to your new data. So you alluded to it and you brought up in the beginning some of your bed rest data and how some of the similarities between muscle disuse or muscle disuse-induced atrophy and cachexia. Do you think some of the neuromuscular junction dysfunction that you're seeing is preceding weight loss because of some decrease in activity or some sort of fatigue, and which also could also come together with the mitochondrial dysfunction as well?
Yes, I guess, of course, cancer people, they move less because they are not really, due to the disease, they want to stress less themselves. So it is possible that during cancer cachexia, you have also less activity. Maybe it's not like a disuse, and I guess that the two models are a little bit different in terms of signaling cascade downstream, but they can be overlapped a little bit about disuse in the cancer people, and maybe a little bit of mild exercise can help in cancer people, mild exercise, to preserve muscle mass because you avoid some of the signaling.
Yeah, I was going to say, I wonder if increasing activity could maybe delay or offset some of those early things,
yeah.
But thank you very much.
Hey, Marco, it's just really lovely, but I just love the sarcomere, right? So I'm looking at your EM images, and you've got some real variable sarcomere lengths in there, and that would suggest you're going to have a lot of variability in titin length. And so I hate to ask you, but have you done any titin gels to look at whether you've got changes in splicing and that kind of thing? Because that has now become a real important part in setting sarcomere length.
I always would like to look at titin. I try to convince titin people because it's such a giant protein that it's difficult to have the test, they say, to monitor if it's fragmented, if it's altered or not. So I don't have the question. I would like the titin people working on titin would help us in addressing this point. But the nebulin is one of the proteins affected. Okay. Okay. Thank you.
Thanks for the great presentation. Earlier you showed that the overall knockout showed the dramatic changes in body weight and survival rates, and earlier you showed that OPA1 was dysregulated in cachexia, cancer cachexia. Have you done any OPA1 overexpressed mouse in cancer cachexia?
No.
Are you planning to?
We did some overexpression OPA1 overexpressing mine in the denervation, so in muscle that are denervated, and you have a beneficial effect. So when you overexpress OPA1 in disuse, like denervation, they have a downregulation of mitochondria, then you have to express a little bit 2-3-fold more, because if you have more than 2-3, it can be toxic. But in condition of denervation, if you have twofold increase in OPA1, you do prevent or reduce muscle loss.
Thank you.
But I haven't tested in cancer cachexia condition.
Yep.
Thanks, Marco. Do you think all your noggin is muscle-derived, or do you think they're, particularly in cancer, is a different cell source?
So, good question. We are doing a single nucleus RNA-seq to determine which are the cells that can produce. I can a little bit anticipate that indeed there are some noggin and other BMP inhibitors are in the myonuclei and are inducing cachexia, but also interstitial cells like FAPs, that they can have a source of noggin.
Do you think maybe fat or tumor too, or really just muscle? Maybe noggin from fat or tumor circulating, or is it really a local muscle environment?
Yeah, the problem is that there are no kit, ELISA kit that can work well in humans. There's only one ELISA kit that works for mouse, and indeed we proved that at least in muscle, noggin protein was induced. We haven't done in the blood in animals, so it is possible that also like a cancer or other organs can produce BMP inhibitors and noggin.
Thanks.
I can exclude.
Thank you very much. Okay, so Dr. Sue Bodine is our next speaker. Dr. Bodine's talk is entitled Disuse Atrophy, Mechanisms in Adult and Old Animals.
So first, I'd like to thank Stu for putting together this session, and also the organizers of this IBEC meeting for putting together a great meeting. It's great to see everybody in person again. So these are my disclosures. So today I'm going to talk about skeletal muscle atrophy, and Marco gave a great introduction, and there's going to be some interesting overlap between our talks, which is great. So we all know that muscle atrophy can be caused by a number of different triggers, and they're outlined here: neural inactivity, decreased loading, decreased nutrients, increased cytokines, increased oxidative stress, glucocorticoid excess, metabolic stress. And over the last 20 years, a significant amount of research has begun to identify all the many pathways involved in these different atrophy processes. And I think one of the things I want to emphasize is atrophy is not a single disease. It's triggered by these different, or it's induced by these different triggers, and different pathways are activated or inhibited. Many of these pathways then are impinging on two important processes: protein synthesis and protein degradation. And the question is always, and some of the debate is, to what extent is protein synthesis inhibited, and to what extent is protein degradation activated? And the net balance with atrophy is generally then an increase in degradation or catabolism versus anabolism. And so today I want to talk a little bit about this balance between synthesis and degradation with disuse atrophy in particular. Also, we've known that in 2021 we identified MRF and MAFBACs as two E3 ligases that go up with muscle atrophy. And so these have become, they were initially identified in a mobilization in the rat, and then have subsequently been identified to be upregulated in multiple conditions that induce atrophy. But one of the big questions still remains is, are these good surrogates markers of protein degradation? They're excellent markers of protein, or of muscle atrophy, but are they surrogate markers of protein degradation? And so I'd like to, at the end of my talk, the second half, talk a little bit about some of our recent work with MRF and determining whether or not is a good marker of protein degradation. So we've used the hind limb unloading reloading model in the rat to look at the effects of disuse atrophy and then the effects of reloading. Today I'm going to talk mostly about the disuse component. We've been using the Fisher-Brown Norway rat from the National Institute of Aging's repository and looking at using adult that are 9 months of age and 28-month-old old animals. And so this is the response of the soleus, the plantaris, and the tibialis anterior to disuse, to 14 days of disuse, and this is absolute muscle mass, so you can see the significant loss of mass in all of these muscles, but greatest loss in the soleus. And then upon reloading, you get recovery of muscle mass and return to baseline for most of these muscles within 14 days. I want to point out this initial edema we see at 1 day, which is prominent in these large mixed muscles, but gets resorbed and then you get progressive increase. Because I'll point this out because with its relevance to aging. But so we've looked at protein synthesis in this model using puromycin, and so this is the loss of or measurement of protein synthesis over 14 days of unloading. In the soleus, medial gastroc, and TA, you can see the soleus has the largest, greatest percent decrease in mass, and also has the greatest suppression of protein synthesis here, which is continued suppression over the 14 days. Less or similar initial degree of drop in protein synthesis in the medial gastroc, but recovers over the 14 days, and less of a decrease in protein synthesis in the TA, which recovers to baseline at 14 days. If we look at markers of protein degradation, so this is the 26S proteasome activity, we see that increases in proteasome only in the soleus muscle and no increases in either the MG or the TA. And then looking at cathepsin L, we see again significant increases in cathepsin L activity in the soleus, but not in the TA. If we then look at MRF and MAFBOX expression over the unloading period and also reloading period, we see that in all of these muscles, MRF goes up, peaks around 7 days of unloading, and then comes back down to baseline. And interesting, both MRF and MAFBOX have this response to loading is an actual suppression or repression below baseline in response to loading. But what you see here is the fold increase actually is higher in the medial gastroc than the soleus or the TA, but all of these muscles are increasing MRF and MAFBOX, but they're not all increasing proteasome activity. So while these are exceptional markers of muscle atrophy, they're not necessarily predictive of increased proteasome activity. So in summary, changes in muscle mass are a consequence of net changes in protein synthesis and degradation. The time point matters, and so you can see that as far as what percent, how much of a decrease you're going to see in synthesis or increase in degradation. The response of individual muscles to the same stressor can be variable. It may be dependent on whether the muscle is oxidative or glycolytic or an extensor or flexor muscle. And increases in MRF and MAFBOX expression do not equal increased ubiquitin proteasome activity. So this is then data we have in the old animals and comparing their response to the disuse atrophy and recovery. So the old animals are in the black dots. And you can see in these 28-month-old animals, there's a slight decrease in muscle mass. When we look at cross-sectional area, there's evidence of decreases in specific fiber type cross-sectional area in these muscles, mostly the type 1 and 2As. Here in the soleus in the rat is almost predominantly 90% slow. And in the mixed muscles, the fiber type, the type 2B fibers are the ones showing the greatest loss or decrease in cross-sectional area at this time. But when we look at, just looking at loss of muscle mass over this 14 days, we see comparable amounts of loss of muscle mass in these old animals relative to the adult animals. The big difference here is upon reloading, we don't see recovery of muscle mass in these animals. And you can also see that we've lost this response here, the edema upon reloading, and in fact, we see a decrease in mass at these early time points in these large mixed muscles. So the other thing that we've noticed is we measured strength in these animals. We can do repeated torque measurements, in vivo torque measurements. This is looking at baseline and then 14 days of reloading in the young. But what you see in the old animals, is a considerably greater decrease in muscle force or in torque measurements, even though they have similar degrees of atrophy. And so we've been interested in trying to understand what's responsible for this large decrease in muscle, in torque or strength, voluntary strength, or nerve-induced strength measurements in these old animals. So we've looked at protein synthesis in response to the unloading period, and you can see here, all of the colored bars or colored symbols are the old animals. So the soleus, there's a similar decrease in synthesis immediately, and then somewhat less or better a recovery over the 14 days. Medial gastroc, an immediate decrease, but then recovery of protein synthesis by 7 days, and no decrease in protein synthesis in the tibialis anterior. When we look at proteasome activity, this is 20S and 26S beta 5. Again, the symbols, the old animals are in the solid. Soleus, now we see no increase in proteasome activity, no increase in proteasome activity in the MG, and a slight increase in the tibialis anterior. When we look at cathepsinel, we do see at baseline high levels of increase in cathepsinel in all of the muscles, and we've measured autophagy in these muscles, and there is at baseline significant increases in autophagy in all of these muscles, but no real change in autophagy or cathepsinel activity over the course of the 14 days. Again, we look at MERF and MAFBOX, so they go up to varying degrees. In some cases, in the soleus, they're somewhat blunted. But again, they're going up, but there's no correlation with their expression levels and proteasome activity. So we've been interested in why we see this similar degree of atrophy in the young and old animals, but why are we seeing this large decrease in force? And so are the mechanisms responsible for the atrophy in these old animals similar to the mechanisms that we see in the young animals? And so this is just a summary of some of our data that's been published, but at baseline, we actually see increased levels of protein synthesis at baseline, and I'll show you some of this data. We see no increase in proteasome activity, and as I showed with the disuse, and we also see this with recovery, we actually see suppression of proteasome activity. But we do see baseline elevated levels of autophagy, but that level of autophagy doesn't change over the course of the unloading and the reloading. This is leading to we're seeing increases in protein aggregates, increase in P62 expression, and increases in the unfolded protein response and AR stress, all suggesting we're having altered proteostasis in these muscles at baseline. And so this is some of our data that we've collected with Ben Miller. We used deuterated water, labeling for 14 days at baseline to measure baseline protein synthesis, and then he fractionated looking at myofibrillar ribosomal collagen and mitochondria biogenesis. But what you can see here in these three muscles, soleus, plantaris, and TA, is this elevation in baseline protein synthesis in these muscles. We don't see any evidence of suppression of mitochondrial biogenesis or ribosomal biogenesis, at least at baseline in these animals. This is data showing we've measured BIP expression, CHOP expression, and P62. We see significant elevations in all of these markers of the unfolded protein response. ER stress is significantly up and increased P62, which seems to be associated with the increase in aggregates and autophagy in these animals. So we then more recently decided to do an RNA-seq analysis. So we took the medial gastrocnemius of adult 9-month and old 28-month-old animals. This is just looking at the differential gene expression, old versus young. So you see that at baseline there are significant differences in the old, with a large number of genes being upregulated and also downregulated. And those gene, we've looked at the specific pathways that have been downregulated in these animals, and a number of these GO terms are associated with a decrease in vascular genesis and angiogenesis, decrease in axon guidance, postsynaptic membranes, and extracellular matrix genes are all suppressed. And then the genes that are upregulated are related to inflammation, T-cell mediated toxicity, inactivity, and neuromuscular junction instability, and also ion channel activity, and in particular potassium ion channels, or genes associated with activation of potassium channels seem to be activated. We've done some further analysis then to look at what genes are upregulated or changed during the unloading. So this is the young versus the old, and so you can see there's significant number of genes upregulated and downregulated in both young and old, more genes in the young than the old animals. When we look at the overlap, though, what we see is there's a significant number of genes that are uniquely upregulated in the young, and then also uniquely upregulated in the old and not the young, and same with the unloading. And when we look at the recovery, what we see here is this is looking at then those genes that were either upregulated or downregulated in the young animals, and then what percentage of those genes return to baseline within 3 or 7 days. And what you can see is in the young animals, a significant, over 83% of the genes return, both that are up or down regulated, return to normal or to baseline, whereas in the old animals, a significant number of genes are not returning to baseline, and there's a delayed renormalization of these genes. And so we're now in the process of looking at some of these genes, and I want to point out, so here in genes that are in common as far as going up or down. So there are numerous pathways that are similar, but here are some of the pathways that are different in the young and old. One of them is the ubiquitin proteasome pathway, is upregulated in the hind limb unloaded old or young animals, but not the old, and we saw that with our activity measurements. Another one is here, the inflammatory response is suppressed in the young animals, but not in the old, and we do see, we've measured NF-kappa B and other markers of inflammation, which are up in the old animals. There's also a term here, the mitochondria is being suppressed in both the young and the old. But what's interesting in the old animals, there's a delay in the recovery of mitochondria. So there could be some energetic imbalance in these old animals upon recovery. And what we also see is this very high level of protein synthesis. So maybe protein degradation is being suppressed in some ways because there's not enough ATP to support all of both the remodeling process. Just some of the genes that we've looked at, here's acetylcholine receptor alpha and NCAM. So we do see these changes in these genes associated with the neuromuscular junction. You can see that they're elevated in the old animals. Upon unloading, they get significantly induced to a higher level than in the old young, young animals, and then upon reloading, they don't go back down to baseline like they do in the young animals, but actually increase even further, suggesting that we're getting significant potential, at least instability in the neuromuscular junction in these old animals with disuse, which is being further exacerbated in the reloading, and maybe further denervation, which is preventing activation of the strength measurements. Other markers of atrophy that have been, or genes associated with atrophy, GAD45 and P21, you can see they're elevated at baseline. GAD45 goes up to a greater degree with unloading, and again, upon reloading, it gets back to baseline and doesn't recover in the young or old animals. Same thing with P21. With MERF and MAFBOX, we're seeing slightly different. They go up, as I showed you, not as much in the old as the young, but what's different is they don't get suppressed in the old animals upon reloading. And so this may be a significant event here that I'll show you some data that we suggest may support that. So in summary, what we see is altered proteostasis in the old animals at baseline, and also upon recovery. We see similar levels of atrophy in young and old, however, greater loss of force in the old. Suppression of protein synthesis actually during disuse is blunted in the muscles of the old, and there's differential gene expression between the adult and old during the unloading phase, with activation of the ubiquitin proteasome pathway being blunted in the old, which may be deleterious. So now, and then disuse, we think, is disrupting the neuromuscular junction, and especially in the old animals that with aging we're seeing increased instability. So for the last part of my talk, I want to move to MERF and MAFBOX. So as many of you know, MERF and MAFBOX were identified in 2001 as two E3 ligases that are upregulated during atrophy. And you can see upregulated under a variety of atrophy signals. But so MERF and MAFBOX are E3 ligases, which mean they add ubiquitin to proteins. And here's the ubiquitin proteasome pathway. You have the E1 ubiquitin, the ubiquitin being conjugated and then moved to the E2. The E2 associates with the a specific E3, and then the proteins, specific substrates bind to the E3 and then are ubiquinated. The classic is that ubiquination, you get these ubiquitin, these polyubiquinated chains added to the proteins, which then target to degradation for the proteasome. But now what we know is that ubiquination can be a single ubiquitin added to a protein, and that may target the protein for degradation, but it can also just alter the function of that protein. You can also add different chains, and this is dependent on the E2, E3, the specific E2, E3, what type of chains, and that type of chain that is added can either target the protein for degradation or could also alter its function. So the big question has been, what are the substrates for ubiquitin or for MERF1? So one of the first questions we asked was, can MERF1 overexpression alone induce atrophy? And the dogma had been no, that it can't, because based on this one transgenic animal. So we then decided to test this, and so we overexpressed MERF in the tibialis anterior of mice for 7 and 14 days. You can see here that at 7 days, we saw no significant differences in muscle mass. We saw a slight change, although not significant, in cross-sectional area. But by 14 days, we saw significant decreases in muscle mass and also significant decreases in cross-sectional area. Subsequent to this publication and function, Dave Hughes has also worked out the electroporations in the medial and lateral gastrocnemius. And here, if we overexpress MRF in the medial and lateral gastrocnemius, we see significant muscle loss by 7 days. So we see this difference between the TA and the MG, which is also seen in vivo as far as the rates of atrophy and it being different in different muscles. So we do get, we can induce muscle loss, muscle atrophy by overexpressing MRF. So then the question was, what are the substrates? And so we used the electroporation. We saw in these electroporated muscles this small increase in ubiquitin protein or ubiquitin ligated proteins. So we asked the question, is there a way of identifying these proteins? And so in collaboration with Francis Impens and the University of Ghent in Belgium, we took these muscles that had been electroporated with MRF, control plasmid, and then MRF plasmid for 14 days, and then used digly remnant affinity proteomics. So you can take these ubiquinated proteins, digest them with trypsin, and when you digest these proteins, you get these peptides that have this gly tag on them. And then there now been antibodies that have been developed that identify or that bind to this gly tag, and now you can use mass spec to identify those peptides that are targeted or that have been ubiquinated. And so we did this, and what we identified after 14 days of overexpression, we had over 1,000 gly sites that were identified that had increased ubiquination. But of those 1,000 sites, we identified 153 sites that were unique, specifically upregulated in the MRF animals, and 16 sites that were actually downregulated. And these were associated with 45 proteins that had increased ubiquination and 11 proteins that had decreased. And this is the table of all the proteins that were identified. These are the ones that were upregulated and the ones that are down. The highlighted proteins here are ones that in the literature had been reported as putative substrates. So you see the myosin heavy chain and myosin light chain are the two that are most commonly quoted. Titin has been identified as a target of MRF. Here's P62 and glycogen synthase. Obvious is TRIM63 is its autoubiquinase, and then also this deubiquinase. But what you see here, one is there are more than two proteins. There are a number of proteins that are ubiquinated, Titin being the most ubiquinated here with 63 sites, TRIM63 or MRF1 having 16 sites. But the majority of these proteins only have 1 or 2 sites that are ubiquinated. We were interested in the fact that Desmond was identified, and I'll talk a little bit more about Desmond, but there are a number of sarcomeric proteins here, but there are other proteins. There's a number of glycolytic proteins, proteins related to the ubiquitin proteasome pathway, a number of deubiquinases, and also E3 ligases, ERAD, VCP, P62, all of these, and these are 2 substrates for VCP2, or cofactors. So there's a number of different pathways that can potentially be affected by MRF1 upregulation and ubiquination. So one of the things we looked at was, does protein ubiquination lead to the degradation of these substrates? And so we looked in these muscles that were, the mass spec analysis did not show any downregulated or decreased proteins. We went back to the muscles and fractionated, homogenized them and fractionated them. We looked at, here's a couple of troponin T and myosin light chain that were identified in the myofibrillar fraction. You can see here's the quantification, the different time point, the empty vector, the ring finger that doesn't induce atrophy, and then 7, 14, and 30 days of overexpression. So troponin T expression, protein levels don't change. Myosin light chain protein levels do decrease. But these other proteins here, we had VCP, Kelch31, that's another E3 ligase, and UCHL1 is another E3 ligase. All of these proteins actually increased in expression when they were ubiquinated by MRF1. So protein ubiquination didn't equal degradation. As I said, desmin is an interesting target for us. So as you know, desmin's an intermediate filament that links the sarcomere to the sarcolemma. It's important for the structural integrity of the muscle and force production. It's been suggested that the loss of desmin may be initial event that then leads to the disassembly of the myofiber. But the literature suggests that TRIM32 is the E3 that ubiquinates and targets desmin for degradation. But the question is, could ubiquination by desmin being or by MRF be a priming step? And so one of the things we went back and looked at is in our muscles that had been when we overexpressed MRF, so this is overexpression of MRF in the lateral and medial gastroc, we see a decrease in muscle mass of 10% within 7 days. So we then measured isometric torque of the ankle joint and see this significant decrease in force, a much greater decrease in fact than at 7 days relative to mass, which could allude to the fact that the sarcomere is being disassembled. And so we went back to our knockout animals and looked at some of these potential targets. These are wild-type animals following denervation of 3, 7, or 14 days. This is looking at this sarcomeric fraction or the cytoplasmic fraction. And so what you see in wild-type animals, so here's desmin in control, very little desmin in the cytoplasmic fraction. With denervation, you start seeing an increase in the amount of desmin in the cytoplasm. Same thing with myomycin and myosin light chain 2. However, in the knockout, we were unable to detect desmin in the knockout animals in the cytoplasmic fraction with denervation, also no change in myomycin or myosin light chain 2, suggesting the possibility that this ubiquination of desmin by MRF may be setting off the process of disassembly of the myofilament. So this is something that we are now further investigating. So in summary, MRF expression alone can induce muscle atrophy and loss of muscle force. Obviously, further validation of the putative MRF1 targets is required. One of the big unknowns is what is the E2 that's associated with MRF in vivo. There was a recent publication that showed that MRF could ubiquinate titin, and this was all done in vitro with a specific E2, and we looked at the sites that they found that were ubiquinated, and none of them corresponded with what we found. So we think it's related to the E2 that's actually associating with MRF in vivo, and this is still a big unknown. So ubiquination of proteins by MRF alone doesn't seem to target a protein for degradation by the UPS. This could be MRF could act as a priming signal that then leads to further ubiquination by another E3. And there are hundreds of E3s in muscle which have not been investigated, and so further investigation of how these are pairing up is required. And MRF1 ubiquination may serve also to modify the activity or stability of a protein. So with that, I'd like to thank you for your attention and the Iowa Wave, and thank my many collaborators, especially Dave Hughes and Leslie Baer at the University of Iowa, Dave Waddell, who's been a longtime collaborator, Ben Miller at OMRF, and Francis Impens and Liliana Radevicic, who were instrumental in the ubiquination, or the ubiquitin proteomics. And with that, thank you very much for your attention.
Thank you very much, Dr. Bodine. We have time for maybe one quick question, if we have one.
Song Lim, University of Arkansas. Thank you for the nice presentation. I have a simple question. Maybe it's a little bit apart from your topic, but during the muscle atrophy, you show the changes in ubiquitin ligases or lower protein synthesis, something like that. But I was wondering whether, have you looked at protein anabolic suppressors, most likely mTOR suppressor, like Daptor or RED1, changes in those markers during the muscle atrophy?
So is the question, have we used suppressed, if we suppress,
Have you looked at anabolic suppressors, like mTOR suppressors,
Yeah.
like Daptor or RED1 during the muscle atrophy?
so if you give rapamycin to an adult animal, it doesn't result in muscle atrophy. So under resting conditions, mTOR is actually fairly stable. Now, we think, or is suppressed, so mTOR gets activated during growth. So if you suppress mTOR during a growth period, during growth, you will suppress growth.
Okay, yeah, the reason why I'm telling you is that we use different model of muscle atrophy, like disuse and cancer cachexia. We saw changes in those markers, so I was wondering whether you've seen this report. But thank you so much.
All right, thank you. Next up, we have Dr. Troy Hornberger. The title of Dr. Hornberger's talk is Using BONCAT to Visualize Where Newly Synthesized Proteins Accumulate During Skeletal Muscle Growth.
All right, well, like many of you, I've spent the better part of my career trying to understand this basic equation. And in particular, I've tried to understand why this equation works. So why, when you apply mechanical loads to skeletal muscle, you get an increase in muscle mass. And so a lot of my efforts have been aimed at studying the mTORC1 signaling pathway. And so work from my lab, as well as many others in the audience today, have led to this generally held contention that when you apply mechanical loads to muscle, you get activation of signaling through mTORC1. That leads to an increase in the rates of protein synthesis and drives an increase in muscle mass. And so according to this paradigm, if you inhibit signaling through mTORC1 with the drug rapamycin, or if you knock out a key component of mTORC1, such as Raptor, then you should inhibit that increase in protein synthesis and inhibit the mechanically induced increase in muscle mass. But it turns out that's only partly correct. So this is some data we published about 3 years ago, in which we subjected the plantaris muscle of mice to chronic mechanical overload via myotenectomy procedure. So we removed the myotendinous junction of the gastroc. We see an increase in the cross-sectional area of myofibers. If we inducibly knock Raptor out of the skeletal muscle fibers, we can prevent that myotenectomy-induced increase in myofiber size, but we do not prevent the increase in rates of protein synthesis. And we've seen the same thing with the synergist ablation model. So in this case, we removed the bulk of the gastrocnemius, the soleus muscle. Plantaris experiences an even greater mechanical overload, an increase in fiber cross-sectional area. If you treat the animals with rapamycin, you block that increase in myofiber cross-sectional area, but you do not prevent the very robust increase in rates of protein synthesis. So this brought us to a couple questions. One, what is this mTORC1 independent pathway that's promoting an increase in protein synthesis? And secondly, where do all those newly synthesized proteins go if they're not contributing to the increase in fiber cross-sectional area? And so when we started to think of this question, we started to think about, well, where do newly synthesized proteins go during a normal growth response? And ultimately, how does skeletal muscle grow? So we dug into the literature, and we're really surprised to find that there's some major gaps in our understanding of this process. So today I'll talk about some of those gaps in knowledge and how we're trying to fill those gaps in knowledge with a new technique called BONCAT. Okay, so how does skeletal muscle grow? I think the easiest way to approach this is to look at it from a structural level. So we'll first talk about what happens at the macroscopic level, then the microscopic level, and finally at the ultrastructural level. And I think we can all appreciate at the macroscopic level, you apply mechanical loads of muscle, you get an increase in the cross-sectional area of the muscle. We can see this in humans with resistance exercise, 8 to 16 weeks of resistance exercise, generally giving you about a 5 to 30% increase in whole muscle cross-sectional area. We can see this in animal models of resistance exercise. So this is a weight pulling model we published about a year ago. In this model, we see an increase in the mass of numerous muscles throughout the body. If we take a cross-section from muscles such as the plantaris, we can see about a 20% increase in the cross-sectional area after about 13 weeks of training. And then we have these models of chronic mechanical overload, like the myotonectomy procedure. And with this procedure, we can see a 50% increase in the cross-sectional area of the muscle in just 2 weeks. Okay? So this increase in whole muscle cross-sectional area is a pretty conservative event. How does it happen? Well, it's driven by the changes that occur at the microscopic level. And those changes could be an increase in the cross-sectional area of the myofibers, an increase in the number of myofibers per cross-section, or perhaps a combination of these two events. And I think that many of you will be scratching your head when I argue that C is the correct answer here. And so if you're surprised by that, let me show you some data that supports that conclusion. So if we go to this myotonectomy model, we see about a 50% increase in the cross-sectional area of the muscle. If we look at the average size of the myofibers, cross-sectional area goes up about 20%. If we count the number of myofibers per cross-section, it goes up nearly 30%. So maybe you're thinking, well, this is myotonectomy, chronic mechanical overload. This is not a physiologically relevant stimulus. If you're thinking about that, I'll show you some data from our weight pulling model. So in this case, I'll draw your attention to the triceps brachii. So we see an increase in the mass of this muscle after 13 weeks of training. And if we take a cross-section of individual heads, such as the lateral head of the muscle, we see about a 20% increase in the number of myofibers per cross-section. So this does happen in physiologically relevant models. Okay, so we've got two things going on and two basic questions. One, how does an increase in mechanical loading lead to an increase in the cross-sectional area of the myofibers? And secondly, how does an increase in mechanical loading lead to an increase in the number of myofibers per cross-section? And in my talk today, I'm going to focus on the second question. Okay, so a couple potential mechanisms here. One would be hyperplasia and or myofiber splitting. And in 2019, Kevin Murak published a review article sort of reinvigorated interest in myofiber splitting, in which it was argued that in response to extreme forms of mechanical loading, you can see an increase in the splitting of the myofibers, and perhaps that contributes to the increase in the number of myofibers per cross-section. And I agree with Kevin in that in response to extreme forms of mechanical loading, you can see an increase in myofiber splitting, but I'm not aware of any data which has ever shown that that increase in myofiber splitting can make a substantial contribution to the overall number of myofibers. And if we're going to talk about this topic, I think we have to remain cognizant of the work that was published by people like Philip Goldnick back in the 1980s. For instance, Philip Goldnick performed an experiment in rats in which they did a tenotomy procedure, so very similar to myotonectomy. And to ensure that this was an extreme form of mechanical overload, they had the rats run on a treadmill for 20 minutes per day at a 25-degree incline at over 37 meters per minute. After several weeks of training, they see an 87% increase in the mass of the muscle. They then went and painstakingly counted every single fiber in the muscle, so over 11,000 fibers per muscle. They see no change in the number of myofibers, and when they were counting these fibers, they also measured the number of bifurcated fibers, which is labeled as BF. And they see that that number went from 2 to 5 of the 11,000. So with data like that, it's really hard to imagine that hyperplasia or myofiber splitting is responsible for this increase in the number of myofibers per cross-section. So if it's not that, then what is it? Well, today I'm going to argue that it's longitudinal growth, or more specifically, myofiber lengthening. And in support of that, I'll draw your attention to a geometric model that was published by Leo Maxwell in 1974. And this model was developed in an effort to predict the number of myofibers that would appear per cross-section when architectural properties such as myofiber length, pennation angle, or myofiber diameter were altered. And in one of the examples from this model, it was shown that if you double the length of the myofibers, don't change any other architectural properties, you'd get a 140% increase in the number of myofibers per cross-section. So I think really nicely illustrating that changes in myofiber length can definitely alter the number of myofibers per cross-section. So we said, well, what can we predict with Leo Maxwell's model? If we go back to our results, we see this 28% increase in the number of myofibers per cross-section. So if we plug the architectural properties of the plantaris into Leo Maxwell's equations, what percent change in fiber length would be needed to get that 28% increase in the number of myofibers per cross-section? And it turned out that only a subtle 7% increase in myofiber length would be needed to obtain that change. So we said, okay, this is a mathematical prediction. Does it happen? So we developed a technique for isolating full-length myofibers. We can confirm that they are intact full-length myofibers by searching for the presence of tapered ends at both ends of the myofibers. We measure myofiber length, and we find about a 7% increase in the length of the myofibers. We also were able to measure the number of sarcomeres per myofiber, and we found a similar increase in the number of sarcomeres per myofiber. So with myotenectomy, we're getting an increase in myofiber length, and that's associated with the in-series addition of new sarcomeres. Okay, so maybe you're thinking, well, this is a super physiological model. Does this myofiber lengthening happen, for instance, in human models of resistance exercise? And in response to that, I would first say, well, it's really hard to measure myofiber length in humans. But you can measure a surrogate marker of myofiber length, which is fascicle length. And in 2016, Emma did a review of studies that have measured changes in both muscle size and fascicle length. And when the results from all the studies that have been published were combined, it was found there was a significant positive correlation between these two variables. And more importantly, if you looked at the individual studies that observed a significant increase in muscle size, that was 55 studies, and 26 of those 55 also observed a significant increase in fascicle length. So certainly a number of studies have shown that resistance exercise can lead to an increase in fascicle length and presumably myofiber length. Okay. So how do we get this in-series addition of new sarcomeres? Well, to talk about that, we now have to move to the ultrastructural level and talk about what happens at the level of myofibrils. And there's kind of two schools of thoughts here. One school of thought argues that you add new sarcomeres at the end of myofibrils. So you could add new sarcomeres at one end, you could add new sarcomeres at both ends, but in either case, you're adding the new sarcomeres at the ends of the myofibrils. The other school of thought argues that you add new sarcomeres throughout the length of the myofibrils at various positions along the length. And I've carefully reviewed the literature that's been used to support these different schools of thought. And in my opinion, the strongest data really comes from studies that have concluded that new sarcomeres are added throughout the length of myofibers. And one classic example, this is a study that was published in 1969 by McKay and Harrop. And it's a really simple study. They took rats, they inserted wire markers into the sternoid muscle of rats. That muscle has myofibers that run the entire length of the muscle. So they insert these wire markers at 6 weeks of age, and then they track the position of those markers throughout developmental growth. So the thinking here is that if you're adding new sarcomeres at the end, then the distance between those two, or those markers, would not change as the muscle fibers grow in length. On the other hand, if you're adding new sarcomeres throughout the length of the myofibrils, then the distance between the markers would grow or get bigger as the muscle grew in length. And that's exactly what they observed. So I imagine it's a little bit hard to see these individual wire markers, but this is an animal at 6 weeks of age and out here at 15 weeks of age. And the distance between those markers increased in proportion to the increase in muscle fiber length, suggesting that indeed new sarcomeres are added throughout the length of the myofibers. So how do you add new sarcomeres in the middle of a myofibril? Well, to talk about this, we have to introduce a concept called sphenodes and the transverse splitting of sarcomeres. So if you've not heard of a sphenode before, this is a structure that was originally identified by Heidenhain in 1919. And what Heidenhain observed is when looking at longitudinal sections of muscle, you could find regions where there was a disproportionate number of sarcomeres along the width of the muscle. So in this case, this is a drawing showing 30 sarcomeres at the top, 43 at the bottom. And this disproportionate number of sarcomeres was often associated with the presence of a wedge-shaped structure, which was referred to as the sphenode. And within these wedge-shaped structures, you could find regions where it looked like sarcomeres had kind of just split into two, undergone a transverse splitting event. So the word sphenode appears in the literature for about another 50 years when Schmalbrook makes a study that reports that the presence of sphenodes is very highly prevalent in the muscles from young animals. It becomes very rare in muscles from adults. And so with that, it was proposed that perhaps these sphenode regions represent regions of longitudinal growth. Okay, so after 1968, the word sphenode basically disappears from the literature, but structures that resemble sphenodes do not. So for instance, this is an image that was published by Yu et al. In 2004. They called this a supernumerary sarcomere region, but it's the same concept. So you have this disproportionate change in the number of sarcomeres as you go across the width of the muscle fiber. And interestingly, this is from human muscle, from a human that performed a bout of eccentric contractions 7 days before the muscle was collected. And this study revealed that there was an over 5-fold increase in the appearance of these supernumerary sarcomere regions. And so the authors proposed that perhaps these are regions of longitudinal growth. So we wondered whether you get that same kind of appearance in muscles subjected to myotonectomy. So we collected muscles at 8 days and 16 days after the onset of myotonectomy and counted the number of transverse splits that appear across the width of the myofibers. And what we found was about 11-fold increase after 8 days of myotonectomy, and still a greater than 4-fold increase even after 16 days following the onset of myotonectomy. So myotonectomy really leads to this massive increase in transverse splitting. All right, so how do these transverse splits occur? Well, one proposal is there's this process called H-zone splitting. And in H-zone splitting, you have a parent sarcomere that basically splits into 2 daughter sarcomeres. And in H-zone splitting, the outer half of the daughter sarcomeres contains material from the original parental sarcomere, and the inner half will be newly formed material. The other type of splitting event that's been proposed is called Z-line splitting. So in this case, it's thought you have a weakened Z-line and you form an entirely new sarcomere between that. And if you look through the literature, you'll find examples where it's really hard to tell what kind of splitting event might have happened. So in this case, you might argue that this is an H-zone splitting event, or perhaps it's a Z-line splitting event leading to new sarcomeres here, or maybe Z-line splitting happening here. And this image is interesting to me because it's taken from a soleus muscle that was subjected to synergist ablation. It was published in 1979, and it was thought that this represents a region of weakness in the sarcomeres where you had new stress fibers being formed and then potentially a splitting event taking place. Okay, so with these observations, we formulate a hypothesis, and that is that during longitudinal growth, new in-series sarcomeres are added at sphenodes via a process that involves the transverse splitting of pre-existing sarcomeres. And so the question becomes, well, how can we test this? And that is where the BondCat technology comes into. So BondCat, if you've not heard of this before, stands for bioorthogonal non-canonical amino acid tagging. And with this technique, we can visualize where newly synthesized proteins accumulate during skeletal muscle growth. And so the basic thinking here, the key assumption is that if you're forming new sarcomeres, those new sarcomeres should be highly enriched with newly synthesized proteins. So if these sphenodes contain new sarcomeres, then those sphenodes should be highly enriched with newly synthesized proteins. Likewise, if we can visualize where newly synthesized proteins accumulate, then we could potentially gain insight to the splitting event that led to the formation of those new sarcomeres. For instance, if this was a Z-line splitting event, then we might expect that the stress fibers that were formed would be labeled with newly synthesized proteins, as well as the entirety of the newly formed sarcomere. On the other hand, if this structure was due to an H-zone splitting event, then we would again expect these newly formed stress fibers to be labeled, and then the interior of the daughter sarcomeres. But the exterior of the daughter sarcomeres would not be labeled because they retain the material from the original parental sarcomeres. And this is an image that we recently obtained with the BondCat technique. And so I want to tell you how we got this image, but before I do that, I want to tell you why we're using BondCat as opposed to traditional techniques. And one common traditional technique is the use of radioactive isotopes. So radioactive isotopes have been used to visualize where newly synthesized proteins accumulate, but the spatial resolution that you can get with radioactive isotopes is very poor, and of course, you have the risks associated with the use of radioactive materials. Stable isotopes, often used in humans to monitor the accumulation of newly synthesized proteins, but to the best of my knowledge, there's no way to visualize where those newly synthesized proteins accumulate. The same point is true with heavy water. So heavy water has become a very popular way of monitoring the accumulation of newly synthesized proteins, but to the best of my knowledge, no way to visualize where those accumulate. And then there's finally the sunset technique. It was actually my lab over a decade ago published the first in vivo use of sunset for visualizing rates of protein synthesis. But sunset labels truncated peptides, not full-length proteins, and those truncated peptides would not be expected to accumulate at sites of growth, but are instead probably rapidly targeted for protein degradation. Okay, so BondCat can overcome these issues. And at the core of BondCat is a bioorthogonal reaction, which is a highly specific chemical reaction between chemical pairs that don't exist in natural biological systems. And the most common bioorthogonal reaction is the classic click reaction, in which you have a compound that bears a reactive azide group, a compound that bears a reactive alkyne group, and in the presence of a copper catalyst, you'll form a covalent bond between these two reactive groups. Okay, so it turns out that there are now non-canonical amino acids available that bear these reactive alkyne, or azide groups, and they're all structural analogs of methionine. And so the thinking is that just like methionine, these non-canonical amino acids can be recognized by the methionine tRNA synthase, and then used to charge the methionine tRNA. That methionine tRNA can then deliver the amino acid or the non-canonical amino acid to an actively synthesizing protein. So if you have just methionine around, you'll get these different sites of incorporation for methionine, and if you had a non-canonical amino acid around, sometimes where the methionine should have been incorporated, you'll instead get that non-canonical amino acid incorporated. And so for a minute, I want you to imagine that we have two different types of tissues, tissue with methionine labeling or with ANL labeling. And if we did a click reaction with a fluorescent tag that bears a reactive alkyne group, when we add our copper catalyst to tissues that just have methionine, we'd get no covalent bond formation, because remember, azides do not occur in natural biological systems. But if we do the same thing in tissues that have been labeled with something like ANL, then we'd get covalent bond formation between the ANL and that fluorescent probe, or effectively we get newly synthesized proteins that are covalently bound to a fluorescent probe. And it doesn't have to be a fluorescent probe. So we've done this with nanogold for EM level imaging. We've done it with biotin. And you can effectively tag with anything that has a reactive alkyne group. Okay, so we've done labeling experiments with all these different non-canonical amino acids, and by far we get the best labeling with the use of ANL. But there's one drawback to the use of ANL, and that is, unlike methionine, which fits very nicely into the binding pocket on wild-type methionine tRNA synthase, ANL is just a little bit too big to fit into there. So if you want to do labeling with ANL, you have to use cells or animals that express a mutated form of the methionine tRNA synthase. And that mutation just increases the size of that binding pocket a little bit so that it can now accommodate that bulkier size of ANL. Fortunately, transgenic animals are available, commercially available, that express this mutated form of the methionine tRNA synthase. And the animals that you can get have a lock-stop-lock sequence in front of a coding region that encodes both GFP and that mutated methionine tRNA synthase. And we've crossed these animals with animals that ubiquitously express the Cre recombinase to make animals that we'll just generically refer to as MET-RS mice. And in proof of principle experiments, we fed these animals either chow that contained just methionine, or 50% of the methionine was in the form of ANL and the other half in the form of regular methionine. We put them on this chow for 4 days. We then took a plantaris muscle from each animal, had that pair of plantaris muscles frozen together in OCT. We then take a cross-section of that pair of muscles, put it on a slide, and perform a click reaction with a fluorophore that bears a reactive alkyne group. We then look at those, that pair of muscles, as expected, both express GFP, but only the sample from the animal that had been given the ANL diet has a signal for our fluorescent probe that had the reactive alkyne group. And the signal-to-noise ratio in this instance is really on-off. I mean, we actually have to turn the microscope down, the light intensity, to be able to take images. The labeling is so intense. We can also do single IP injections. So we've injected animals with 100 micrograms of ANL, and we did this after they performed a bout of weight pulling or a control condition. We collected the muscles 3 hours later, homogenized them, and then we performed a click reaction on the muscle lysate, in this case with biotin that bared a reactive alkyne group. And so effectively we get newly synthesized proteins that are covalently bound to biotin. We can perform an anti-biotin Western blot and observe a weight pulling-induced increase in the accumulation of newly synthesized proteins. And when we did this experiment, we also injected animals with PBS as a negative control condition to get a sense of our signal-to-noise ratio. And with this 100 microgram per gram ANL injection, the signal-to-noise ratio was good, but wasn't exceptional. So we next wondered if we give more ANL, could we get a better signal-to-noise ratio? So we injected animals with either 100, 200, or 400 micrograms of ANL per gram body weight. And in a dose-dependent manner, we could increase the amount of ANL labeling that we observed. And with the 400 micrograms per gram body weight injection, the signal-to-noise ratio was really, really good. So having established how to do this labeling of newly synthesized proteins, we now return back to this question of whether these phenodes are regions that contain newly synthesized proteins. And we leveraged off of our observation that at 8 days after the onset of myotenectomy, you have this massive increase in transverse splitting or sphenode formation. And so we designed an experiment in which we took our MET-RS mice, we subjected them to myotenectomy, gave them a single injection of 400 micrograms per gram ANL, and then collected the muscles a day later. We took longitudinal sections and we looked for regions that had a lot of transverse splitting. So unfortunately, it's probably a little hard to see in the back of the room, but this region is just absolutely loaded up with transverse splits. And when we look at the accumulation of newly synthesized proteins, this region is loaded up with newly synthesized proteins. It's at least a 10-fold increase in newly synthesized protein deposition in the region with transverse splits versus regions where there's not. Okay, so this supported the idea that these regions with transverse splits are indeed sites of new sarcomere formation. And so now I want to summarize, and to do that, I'm going to go right back to where I began. So I began by telling you there's this generally accepted paradigm in which mechanical stimuli activate signaling through mTORC1. That promotes an increase in protein synthesis, and it's thought to drive the increase in muscle mass. But then I told you, there's some problems with this model in that when we knock out Raptor, we can prevent the myotenectomy-induced increase in fiber cross-sectional area, but we did not prevent the increase in the rates of protein synthesis. And so that brought us to a couple of questions. One, what is that mTORC1 independent pathway that's driving the increase in protein synthesis? And then secondly, where do all those newly synthesized proteins go if they're not contributing to the increase in fiber cross-sectional area? And so that brought us to the question, well, where do newly synthesized proteins go during a normal growth response? And so we've talked a little bit about how skeletal muscle grows. At the whole muscle level, at the macroscopic level, we can see an increase in cross-sectional area. And at the microscopic level, that's driven by an increase in the cross-sectional area of the individual myofibers, as well as an increase in the number of myofibers per cross-section. Based on mathematical predictions, very small changes in myofiber length could explain that increase in the number of myofibers per cross-section. We've obtained data which supports that you see an increase in myofiber length, as well as an in-series addition of new sarcomeres. And then finally, I've tried to convince you today that those new sarcomeres are added throughout the length of the myofibers at regions called sphenoids through a process that involves the transverse splitting of pre-existing sarcomeres. Okay, so if we go back to this model and try to wrap things up, I think we have to make an additional layer of complication here, because there's really two types of growth. There's an increase in myofiber cross-sectional area, and there's an increase in myofiber length. I've shown you data which indicates that increase in myofiber cross-sectional area is mediated through an mTORC1 dependent mechanism. But what about this increase in myofiber length? Perhaps this is mediated through an mTORC1 independent mechanism. And if it were, then it would explain why we can still see a really big increase in rates of protein synthesis even when signaling through mTORC1 is inhibited. And it turns out that over a decade ago, we published data which supported this idea that the increase in myofiber length is mediated by an mTORC1 independent mechanism. But when we published that data, we didn't really have a good understanding of how skeletal muscle grows, and so we didn't really know what we were looking at. Specifically, I think many of you have probably seen this paper before. In response to synergistic ablation, we saw an increase in the cross-sectional area of myofibers. We treated animals with rapamycin, we blocked that. But when you look at the number of myofibers per cross-section, we did not prevent the increase in the number of myofibers per cross-section. And we've made the same observation with our myotenectomy model. So again, rapamycin blocking the increase in myofiber cross-sectional area, but not the number of myofibers per cross-section. And remember that myofibers per cross-section is a surrogate readout for changes in myofiber length. So if we try to summarize things, what I think is going on is that in response to mechanical loading, we get activation of signaling through mTORC1. That can promote an increase in protein synthesis, and the mTORC1 dependent increase in protein synthesis contributes to the increase in myofiber cross-sectional area. In response to mechanical loading, you can also get an mTORC1 independent increase in protein synthesis. And that mTORC1 independent increase in protein synthesis contributes to the increase in myofiber length. And it's ultimately the combination between an increase in myofiber cross-sectional area and myofiber length that gives us that full growth response. So with that, I hope you feel like you've learned a little bit about how skeletal muscle grows, and you can now appreciate why understanding how skeletal muscle grows can help us gain insight into the mechanisms that ultimately drive that growth response. So with that, I need to send some acknowledgments out. First, I have to thank the funding agencies, in particular NIAMS, for supporting my favorite hobby, which is doing this stuff every day. And I also have to say that the ideas I presented today really represent a culmination of ideas that have been fermenting in my head for over a decade. And so to really send out appropriate level of acknowledgments, I have to acknowledge everybody that I've reeled into the lab during the last decade, because in one way or another, every single one of them contributed to the work that I presented today. So thank you for your time.
Thanks very much, Troy. And I just want to point out that Troy put a few papers in there that were published prior to the internet, and you may actually have to go to the library and use a photocopier or a scanner or whatever it is you kids, use these days. So thanks for that, Troy. Yeah, good physiology was done prior to the internet. Thank you. Questions, please.
Beautiful talk, Troy. I'm guessing you're probably doing this experiment, but did you treat your bond-cap mice with rapamycin and see if they're still newly synthesized proteins? That's the first question. The second question is can you actually use that technology to pull down proteins and identify them and see what's actually being made?
So to your second question, yes. And it seems like it should be simple, but like everything that should be simple, it's always more complex than it's been. So we haven't, we started this bond-cat labeling 5+ years ago. And initially I wanted to really do those pull-down experiments, but we ran into trouble with it, and so we haven't focused too much on that. What is probably not evident in here is we've spent a tremendous of time working out the chemistry to be able to do imaging on tissues that have really exceptionally preserved structure. And I only can show just a few images here, but no, we haven't done the rapamycin experiments. I mean, some of those images I literally collected a week before we got here. So I wanted to share the stuff, so yeah.
Great talk, fascinating to hear. I promised you if you convinced me, I'd come up and say, you convinced me that hyperplasia doesn't exist. But I did want to ask, in relation to, I'm assuming a lot of these pictures were taken closer to the mid-belly, and just your thoughts on what may be occurring at the myotendinous junction, as well as sarcomeres in series versus in parallel, and where we may be getting some of these changes related to strength, CSA, and all of that.
Yeah, so a couple things. So I think there is heterogeneity along the length of the muscle in terms of what types of growth you get. And there's a lot of papers that have been published on this heterogeneity. I think, Stu, you probably published some of them yourself. So there's heterogeneity. We see those hot spots, those phenode regions along the length of the muscle. There's no prevalence to distal end or proximal end. The images, the one image I showed is just a randomly selected region, but we see them all throughout the length of the muscle. So, yeah.
This is going to have to be the last question, a little bit behind.
Thanks, Troy. That was fantastic. I really enjoyed that. Have you, just following up again on the first question a little bit more on identifying proteins, do you think this method could start to tease apart sort of translational mechanisms of protein synthesis, like cap-dependent, independent, things like that, and how mTORC1 might drive that?
Yeah, so some of the delusions of grandeur that I had at the beginning when we were doing this was we would do about a weight point, for instance, and we could click on biotin, pull down biotin labeled newly synthesized proteins, and see what proteins are being formed versus that are not, or that are showing increase in rates of protein synthesis. Those experiments are entirely doable. We just don't have the funding, haven't had the funding or the manpower to do it, especially with COVID. So,
well, if you want to do some mass spec, maybe we should team up and, yeah, I'd be happy to.
Yeah. Well, so as you know, we do a lot of work with Josh Kuhn, and so we were completely in line to do a lot of this stuff, but it's been trickier to do than we expected, and we don't have enough funding. So.
Thanks.
All right, then apologies to cut off the questioning, but there is a lunch break after this, and so you can prevent Troy from getting his lunch and ask him questions a little bit later. So thanks, Troy.
Okay, so last and hopefully not least, but here we go. So I'm going to talk about what we've learned in terms of gaining muscle and what we might use from that to try and prevent muscle loss during atrophy. So these are some of the funding agencies that have supported the work that we do in the lab, and these are some companies that have paid honoraria to me and also that I have shares in, and that's Exerkine. This is the last time we were able to gather. You can see we didn't have masks on, but it was only for the 3 seconds to take the picture, and we rushed to get our masks back on after that. but according to the Government of Ontario, we were allowed to congregate, and that was the maximum amount of people that we could have in my backyard at the time. These are the people that do the work, so I just really want to acknowledge them. There are a couple of great references that I just want to tap that are, I think, pretty good in terms of reading, and I'll blow our horn here. Everson Nunez is a postdoc in my lab. We recently published this paper. This was a really great write-up after a symposium and I think one of the last experimental biology meetings that is a good read on this topic. So I'm going to talk about simple disuse atrophy. And it's induced by unloading, either by a bed rest model or limb immobilization, and it has a number of characteristics. We can also induce it using relative unloading, or abrupt sedentarism, or hypodynamia. And you'll recognize that that's probably a characteristic of a number of muscle wasting disorders, but it is not a model that's accompanied by hypercortisolemia, hypercytokinemia, and what it is not is it's not cancer, it's not AIDS, it's not sepsis, uremia, or uncontrolled diabetes, or cachexia that you heard Professor Sandri talk about this morning. This is simply the removal of the mechanical loading stimulus. You've probably seen these types of figures before if you've heard me speak. You've probably seen some of them in our papers, and it really describes the vacillation between fasting and fed states of muscle protein synthesis with a wandering sort of small change in proteolysis, and that's really what describes the normal anabolic state in response to protein ingestion. Things change a little bit when you perform resistance exercise or loading, because what you get now is a sensitized or increased anabolic effect and a reduced catabolic effect because of the predominant stimulation of muscle protein synthesis. Again, breakdown really doesn't change too much. If anything, in this model, it actually goes up a little bit. So the main question then is what happens with decreased loading? So I've got somebody with a crutch here, somebody on bed rest, and I suppose the situation is that you could get a reduction in synthesis, this so-called anabolic blunting, which would be one mechanism. Or you could have a situation where you're talking about breakdown being increased. And so it would look probably something like this, again, to exaggerate the effect. And the answer to the question has been a little bit elusive. It's been difficult to tease out. And one of the reasons why it's so important is that the likely choice then of therapeutics, or what you're going to focus on, would depend on which process is predominant during the catabolic condition. So what happens to muscle protein synthesis and disuse atrophy in the fasted state? And again, just to prove to you that good science was done before the internet, this is Mike Rennie. I couldn't find a picture of John Gibson, unfortunately, but this is work that he did with Mike Rennie looking at men, and it was all men, who fractured a limb after a motorcycle accident. No women doing that. There you go. Not a comment, just the way it is. And this is the uninjured versus the injured limb, and this is the protein fractional synthetic rate in one limb that is wearing a full cast. So this was a tibial fracture. And I think it's pretty clear that you see a reduction in the injured limb. That's a very reproducible observation. This is in the fasted state, but these are papers. This is actually, I don't know it's even an exhaustive selection, but it's something that comes up time and time again, and you'll see it again several times throughout the talk here. So it's something that's very reproducible. Protein synthesis goes down in the fasted state. So the question then is what happens in disuse atrophy in the fed state? And in this situation, and this is Eliza Glover, who was a PhD student at the time in my lab, and we infused amino acids either at a low dose, this would be sort of a small meal, about maybe 10 grams of protein, and this would be a very large meal, closer to about 50 or maybe even 60 grams. And what you can see in a non-immobilized versus the immobilized limb here is a drastic reduction across 2 and 4 hours of incorporation in protein synthesis, and it doesn't matter whether it's low or high dose, and you can already begin to see this separation at the fasted condition here. And so clearly when we feed people, you're getting a less anabolic response in result as a result of hyperaminoacidemia. And just to prove to you again that it's not just us, this is Ben Wall. He was working with Luc Van Loon, good friend and colleague, and he showed again that this is the resting fasted state synthetic rate. It's down, and this is the fed state in response to actual real food ingestion, so not a hyperaminoacidemic sort of clamp situation. So again, this seems to be a fairly reproducible observation. One of the comments that we get is you're not measuring breakdown, and that's true. I'll freely admit, so you default to synthesis, and people say, well, it's more technically demanding, and that's true. It's absolutely true, and I'll say that unequivocally. It's much more difficult to measure protein breakdown using either stable isotopes or any methods out there. So people have looked at indirect snapshot type markers to infer that proteolysis is up. And I'll maybe hopefully try and convince you that breakdown has been measured in simple disuse atrophy models. And when it has been measured, it actually doesn't change too much. So breakdown, and you would think it should go up. But remember, this is a simple disuse atrophy model. It's not a model of, I guess, precipitous or very rapid muscle loss. So this is the paper that most people refer to, and this is Pertesh, and this is interstitial. So this was using microdialysis, and you can see an increase in 3-methylhistidine. For those of you who remember, 3-methylhistidine is a post-translational modification of histidine residues in actomyosin proteins, and lacking a tRNA, once the proteins are degraded, it's unable to be recycled. So we used to put people on meat-free diets, and we used to quantitatively collect their urine, because this is excreted in the urine, and say, this is a marker of proteolysis. However, the observation is that, you know what, maybe we don't know where those actomyosin proteins are coming from, but Per and his group here did a nice job in putting a microdialysis needle through the thigh of muscle of individuals who were, this was only 3 days of immobilization. And it's the piece of data that most people point to as saying, look, maybe there is an increase in proteolysis, and okay, I would buy that. Maybe it's only an early event, so it's 3 days in. So just bear that in mind as we move forward. This is an interesting model. For those of you who don't know, this is Vince Chiesa, and Vince and his group down at UC Irvine were really interested in how we can offset atrophy that might occur during spaceflight. So this is a really cool paper, actually. It was a really cool model. So they've got vastus lateralis and soleus biopsies from these individuals, and they've got them on bed rest, or AG stands for artificial gravity. So what they did in the AG group was they actually got them out of bed, and they had them pedal in a device that the faster you pedal, it went around, and essentially you became like, it was like a human self-powered centrifuge. And yeah, it was pretty cool. And what you can see is that there's a reduction, didn't quite achieve statistical significance in the vastus lateralis, but definitely in the soleus, that is completely offset in the group that is able to subject themselves to an artificial gravity, so essentially spin themselves around and generate a small gravitational field. But you don't need much, as you can see here, to offset the effects of this, this negative stimulus here. Interestingly enough, biopsies from this group were taken, and measurements of muscle protein synthesis and muscle protein breakdown were done. And this is Brock Simons, who was, I think, a PhD student at the time with Doug Patton-Jones. And what you can see is this stereotypical, this is the artificial gravity in the bed rest group before and then after, and not surprisingly, bed rest, there's the decline in fasted state muscle protein synthesis. This is the artificial gravity group here. No real change. In fact, so a difference in basal muscle protein synthesis. But you can see proteolysis doesn't really change much. So, you know, this at least suggests that the predominant mechanism that's accounting for the atrophy is a reduction in muscle protein synthesis. I'm going to take you back a little bit, and this is some data that Mark Tarnopolsky and I collaborated on to generate. I think it's some of the first, and it was gene chips when gene chips were cool. Now it's all RNA-seq, but I wanted to show you that this data actually holds up pretty well. So again, this is Elisa Glover. I couldn't find a picture of Nobuo Yasuda, who was a PhD student of Mark's at the time, but he and Elisa collaborated on this investigation. It's pretty cool because we did this in men and women. We had equal numbers in each group, but this is just really to show you that our immobilization model worked. In other words, we got a reduction in muscle fiber cross-sectional area, and in muscle cross-sectional area, it didn't really matter what we looked at. And the women lost about, relatively speaking, the same amount as the men. One of the interesting observations is that the women lost quite a bit more strength than the men, which is something that nobody to date, at least, has followed up on. So the model works, and when we look at the chip measured changes, you'll notice lots of different pathways. This is sort of a gene ontology type pathway analysis. And you'll notice that one of the most profound reductions that we get is the reduction in the amount of mitochondrial transcripts. This is only 2 days into the unloading. We did it for 14 days, and this is what happens at 14 days. So actually everything else sort of settles down, but you're still getting a very, very profound reduction in mitochondria-related transcripts. And I didn't go into the gene level or gene protein level at the time because some of the software that you have now wasn't available, but just to show you that this signature is quite pronounced, and it's probably one of the key events that is triggering the reduction in protein synthesis with immobilization. We did measure atrogen and MRF protein and gene abundances. This is the gene transcript level. We also measured the proteins, but just to show you, we do get some increases, not as pronounced as you might see in some of the rodent models, and the proteins were actually a little bit muted even compared to the genes themselves. So one of the questions we asked is, well, maybe hypertrophy and atrophy are simply the opposite of each other. And, so if protein synthesis underlies the changes in hypertrophy and atrophy is the diminution of protein synthesis, then can we develop a model to look at these and sort of decomplicate it a little bit, because the between-person variability in humans is profound, and it introduces a lot of complicating factors when you're trying to figure out what is responsible for one or another of the processes. So Tanner Stokes, who's right on the cusp of defending his PhD in my lab, developed this, what we call the HIPAT model. So we had 10 weeks of unilateral resistance exercise, 3 days a week, which we knew stereotypically on average would give us about a 5 to 7% increase in mid-thigh muscle cross-sectional area. And length, I'm assuming too, Troy. So, and we 2 weeks only of immobilization. Ethics wouldn't let us immobilize people for 10 weeks because there wouldn't be much left of their leg at the end. But 2 weeks of immobilization is about the equivalent of 10 weeks of resistance training in terms of what goes up must come down. So it takes 5 times as long to grow as it does to lose the muscle. So bear that in mind. We made measurements with ultrasound, and we took MRI images at the beginning and the end here just to make sure that we were getting atrophy in this leg and nothing was changing with respect to the opposite leg here. So we've got one leg growing, one leg shrinking at the same time. The first cool part was, the good news is ultrasound does a really good job. If you're a skilled operator and you can hold the probe steady and you're very religious about how much gel you put on, don't apply any pressure, you don't have to put people in an MRI magnet, which was good news because it's pretty expensive. So there was a great correlation between the percent change up or down and the percent change measured by ultrasound and MRI, and it didn't matter, whether you measured this in hypertrophy or atrophy going in the positive or negative direction. And you can see that in the paper that Tanner published there. The interesting part, again, a lot of people want to know what happened to the immobilized limb. Remember, it's not immobilized for weeks 0 to 8, only in the last 2 weeks. Was there any effect of the resistance exercise on this leg? The answer is unequivocally no. It doesn't change. And so it's a good control then for the initiation of the 2 weeks of immobilization leading to atrophy here. So about a 5 to 7% increase up and about a 5 to 7% decrease down. So can this HiPAT model then improve our detection of some of the molecular regulators of muscle remodeling? And so we collaborated with Phil Atherton's group to measure muscle protein synthesis using deuterated water incorporation. And we looked at microarray analysis, and we chose to use microarray for a number of reasons. I know RNA-seq is the flavor of the day. The interesting part is that RNA-seq doesn't give you any information about the regulation of genes that might occur at the untranslated regions, either 5' or 3'. It's just the transcript itself. So you're going to miss some of the molecularly regulated events if you choose to use RNA-seq and you don't use microarray. When we did this, 1,435 transcripts up, 649 down, and if you look at how many were regulated at the 5' and the 3' end, these are data that you would have missed had you used RNA-seq. So we think we've got something pretty unique when we looked at this, and what we did with this was then to create, using our 14 individuals here and within-person variability, which is an advantage of the model, a signature that related to muscle gain using independent verification from another 100 subjects that we had from previous studies that we hadn't done anything with in terms of the data. So this required a lot of patience because we sat on these data for some time. But what we found was there was about 141 growth correlated transcripts, and 50% of them were regulated at their untranslated regions. This is a model that is derived through machine learning and artificial intelligence. It's completely unbiased. So the machine does all the learning. And what it does is then generates a list of pathways and genes that are associated with them. And it's probably no surprise that we come back with a model of these 141 transcripts that probably relate to pathways that would surprise nobody in this room. So we thought, we're onto something here. The reviewers at Cell Reports weren't convinced. If you're out there, I hope we convinced you because we got the paper published. But what we did to show that they were important genes was selected a few of them and then collaborated again with Hannah Crosland, who was a postdoc at the time working in Phil Atherton's lab. And we used microRNAs, inhibitory RNAs to knock down the genes of a few of the proteins, a few of the genes that we said were related to muscle growth. And when we do that, and in cells and culture, when they were challenged with an anabolic stimulus that you can see, this is the puromycin incorporation here, BCAT2 and FKBP1. You can see that first we knock the genes down. I haven't shown you that figure. But when we do that, you can see that there actually is a functional outcome associated with a reduction in the expression of these genes, and that's an inhibition of protein synthesis. We've done this for several other genes as well, and the results are broadly speaking similar. So we think that we're onto something with this gene signature. I'll just show you this because the reductions in integrated myofibrillar protein synthesis that we determined across the 2 weeks of the immobilization phase were related to the change in vastus lateralis cross-sectional area determined in the unloading phase. And so remember, we didn't have the MRI measures here, but we did have the ultrasound measures. And interestingly enough, the reduction in integrated myofibrillar protein synthesis related to the degree of change in the vastus lateralis cross-sectional area that we measure with ultrasound. So again, hinting that this is the predominant adaptation that is responsible for the atrophy. One of the questions, remember, the Pertesh data is, does this happen over the short term? So again, we worked with Phil Atherton, and this is Matt Brook and Tanner worked on this study together, and Stefan Gorson, who was a postdoc at the time, helped us out as well. We had a short-term immobilization protocol, only 3 days in duration. We had DEXA and ultrasound strength. We did deuterated water. These are all of the sample. And then we had an acute infusion trial. And the acute infusion trial was a little bit, and you'll forgive me here, for this is measuring protein synthesis, but we had these 2 stable isotope injection pulse tracer type things that allow us to make an instantaneous calculation of protein synthesis. It's pretty tricky, but I think you'll see we got some data which, at least in our hands, we think makes sense. We initiated a hyperaminoacidemia, hyperinsulinemia, hyperglycemia by simply infusing amino acids and dextrose. Took lots of blood samples and then muscle biopsies from both legs. So this is the rate of muscle protein synthesis. Again, this stereotypical, remember this is the integrated rate across the 3 days. Reduction in the, from the control versus the immobilized limb. This is the fractional growth rate, which you can compute from taking the actual muscle mass that's there, same type of findings. And this is the correlation between muscle protein synthesis and the change in muscle thickness. And you can see we sort of get a similar type of relationship. I hasten to add that this came out just in time for the conference, so I can talk about it without the feeling that somebody's going to take a picture, put it on Twitter, and scoop us. But there you go. When we tried to measure protein breakdown, this is the indirect method of measuring fractional breakdown rate. So you would have to sort of go back to measuring the protein synthesis and then taking the difference by calculating the mass of the muscle. So obviously not a lot of noise in there, but something that we couldn't detect changes in protein breakdown. When we measured it using the pulse tracer approach here, again, you'll notice that we lost a few samples. We were running really shy on tissue here. We don't see anything that at least convinces us that there's an up or a big down regulation of muscle fractional breakdown rate. So we worked again with Craig Willis. I see his name's come up here with Dr. Bodine's work as well. He really helped us with the RNA-seq analysis in this model. And what Craig did was essentially identify pathways that were associated with protein synthesis and with the change in protein breakdown and the change of muscle mass. When we did that, there are some key hub genes. This is Deptor, so these were of undetermined function, but possibly related to muscle protein synthesis. These are some of the potential transcription factors that interact with these proteins here, and the size of the circle is essentially the weighting that the model gave to some of these genes. And then there are things that are related to ubiquitin protein transferase activity here, and you can probably pick out some of your favorite genes here, and then some of them that we have no idea what they do. So what can we do to mitigate disuse? And I'm going to highlight some of the work that Chris McGlory, who is a former postdoc, now a faculty member at Queen's University, essentially did using omega-3 fatty acids. So these are very high doses of omega-3 fatty acids that we delivered to young women who went through a 2-week immobilization protocol. You probably, well, you definitely have to use supplements to do this, or you'd have to eat a lot of fish. I don't even think the Scandinavians could eat this much fish. But this is to show you that this is the peak quadriceps cross-sectional area, and I could use lots of different measures here in the 2 weeks of immobilization and then 2 weeks of recovery in an omega-3 supplemented group versus a placebo supplemented group. So you can see it's attenuated in the omega-3 group. It's by no means inhibited, but this might be important because the valley is lower, and then when you get into recovery, you'll see that these women actually are probably in a better shape. Just again to show you, these are the DEXA data. I know it's not the greatest measure, but if you look at this, you can see that and this is maybe suggestive that it's not until the latter half of the immobilization period where you begin to see changes in the omega-3 supplemented group. But you can see if this were your measure here, then by 2 weeks of recovery, these women are back to where they started. We collaborated with Graham Holloway. This is Paula Mioto, who graciously drove up and down Highway 6 to grab the muscle and then run back to her lab and make measurements of mitochondrial respiration. What you can see, we took biopsies early in the process, so in the first 3 days and then in the post-immobilization. In the control group, and this is an ADP titration curve, you get a large reduction in mitochondrial respiration. What you don't see is that in the omega-3 supplemented group. So we think that targeting mitochondria in this situation might be one of the key reasons why this intervention worked as well as it did. Just to show you that it's not just omega-3s, this is, I think, one of the few papers, it doesn't get cited an awful lot, but highlights some really hard work that Tanya Holloway was doing in my lab when she was a postdoc. And using a specific amino acid formulation that was developed by a startup company in Boston called Excella, so it's AXA 2678. You can see its formulation there, and we delivered this 3 times a day. By my calculations off the cuff, it's the equivalent of eating about an extra 100 grams of protein per day. You could try that if you like. It doesn't have N-acetylcysteine in it, which I think is a key ingredient, but I think this is really only practical using essential amino acid formulations. Just to show you, this is the mid-thigh cross-sectional area. This is the decline in the AXA-treated group versus the placebo across about a week of immobilization. And you can see the same in muscle volume that was determined by MRI. This is the integrated area here. So this is the placebo, the AXA-treated group, and the same with muscle volume. So if you give enough of these essential amino acids, you can possibly blunt some of the changes. This is short term. It's 7 days. So in other situations, I'm not so sure. So some take-home points. First of all, changes in muscle protein synthesis mediate changes in muscle mass. It's something that's obviously affected by protein and loading. I didn't show you the data, but clearly it's also affected by aging. In fact, the immobilization effect and that anabolic resistance that occurs is stereotypical of what happens in older individuals as well. Simple disuse results in declines in muscle protein synthesis in both the fasted and fed state, which I think, if you do the math, can account for the majority of muscle loss. So I'm not sure that you have to really invoke changes in muscle protein breakdown. So, and in most human studies in which muscle protein breakdown has been measured, we don't see a profound increase, even in the short term, and that's despite the indirect, you'll recall, dialysis markers of 3-methylhistidine. Omega-3 fatty acids in very high doses or high-dose essential amino acids, or possibly other mitochondrial target therapeutics might help. But I think that it's probably the link between these molecular pathways and mitochondria that are preventing this decline in muscle protein synthesis. And so possibly through mitochondrial metabolism being effective, and I think that should probably be the top therapeutic target in a simple disuse atrophy model. When you layer on top other hypercortisolemia or hypercytokinemia, a massive inflammatory state, a lot of this, I'm not sure it would work. So you would have to target the inflammation or the hypercortisolemia first before some of this would work. Thank you very much. Hopefully there's time for a few questions. I know I'm standing between you and lunch and we're already late, so I'm happy to entertain questions if everybody else wants to take off. So thank you. No questions? Perfect. Okay, everybody, enjoy the conference. Posters across there. Thank all the exhibitors outside, and enjoy your lunch. Take care.